Okay, welcome back. If you can please settle down, we can move on to the next presentation. So it is a pleasure to welcome and introduce Dr. Deng, who is our second local representative here from the Les Turner Foundation. Can you hear me? Oh, I'm sorry. How about now? Better, okay, great. So I was saying, welcome back. It is a real pleasure to welcome and introduce Dr. Deng, who is the second of the local representatives here from the ALS Center. So Dr. Deng is a professor of neurology at our department. He received training in clinical medicine and research in China and Japan. And his research focuses on identifying genetic causes of diseases of ALS and elucidating underlying mechanisms. Dr. Deng has really been the driving force in Tipu Siddique's lab for about 20 years, and he was behind a number of important discoveries, including the identification of several ALS causal genes that developed on several mouse models. And in his own independent program, he's continuing along those lines. And today he's going to talk to us about ALS5, SPG11. So welcome, thank you.
I don't know where did you find my my early pictures? Those young. How can I handle this one? Where are they?
Big green arrow here.
Big green. Is there any lighter? So I can point it.
Right here, this is the laser. There you
Okay, that's good. Thank you. So today I'm going to talk about the ALS5, and which has not been reported before. And ALS5 is a juvenile ALS, meaning the disease onset is very early. Normally, we say ALS is adult onset, 50, 60 years, mostly. And ALS5 is very young. So they develop a disease before, we say the onset is before 25 years old. So called juvenile ALS. This disease progression is very slow. So that takes like the disease course like about 30 to 40 years. And so, initially they develop so-called spastic paraplegia, meaning the upper motor neuron degeneration. So that's why in the early stage, the disease will be diagnosed as SPG, spastic paraplegia. But disease will progress. Eventually, they will involve, mostly after 10 years of onset, they will develop low motor neuron disease. So at this point, patient will be diagnosed as ALS. So it's the same disease, just a different stage when the doctors examine the patient. Eventually, they may develop other symptoms. For example, not only upper motor neuron and low motor neuron, but also other neuron systems. For example, dopaminergic neuron, then patient will be diagnosed as Parkinson's disease. If they have sensory problem, they will diagnose like Charcot-Marie-Tooth disease, both motor and sensory. So it's a very complex disease. That's why we call it, in spastic paraplegia, they called complicated spastic paraplegia. So the clinical diagnosis is very complicated. And in 1998, Dr. Han-Tati, then working in Dr. Siddique's lab, mapped this gene to this region, chromosome 15, and about 8 centimorgan region. 1 year later, there's a European group, and 2 years later, there's a Japanese group. They mapped a gene called spastic paraplegia to this region. So this region is much larger. And in this region, in our region, there's 120 genes. And we sequenced 51 genes. We couldn't find anything positive, meaning cause of disease. And in 2008, 7. And Dr. Srivani identified a gene at that time called KIAA 1840. It's a disease caution. Then they turned this gene called SPG11 because they cause disease called SPG11, spastic atresia type 11. And later on, there's so many different diseases. They're called juvenile ALS, reported, also caused by this gene, and Charcot-Marie-Tooth disease also caused by this mutation of this gene, and Parkinson's disease caused by this gene. That's why previously I said early they may be diagnosed with spastic paraplegia, and later on there are so many, they involve so many areas, so many types of neurons, so they will cause, clinically, they will cause different disease, but predominantly is motor system. This patient, they also cause disease. They called retinal atrophy and ataxia, and more than 50% of patients, they will develop ataxia. So clinically, it's a very complex disorder. So they can cause the other, for example, they cause the cognitive impairment. Patients eventually they will become impaired, cognitively impaired, and they will die normally, mostly before 60 years old. And because this gene is identified, like it's called SPG11, and because this patient also, more than 50% of patients, after 10 years, they will develop ALS. So we thought ALS and SPG11 maybe just the same disease. So then we sequenced our patients with we called ALS5. So we sequenced these three original families, which we used to map this disease gene. Actually, we found all of them have the mutation SPG11. And we also found these 3 families is used initially by Dr. Siddique's lab. And then we found a patient in North America, and we found 3 families, 4 mutations, also caused by, when the patient is later stage, they develop ALS. And what is the problem with this gene? What is the function of this gene? And we had two the autopsy samples from 2 ALS patients, and what we can see is this is a normal people without neurological disease. These 2 are ALS5 patients. What we can see is this is the brain, and then we can see this area is we call the gray area. This is meaning the neuronal rich area, and this white area we call the white matter. These are external area, meaning the neurons have axon extension, and the neurons are here, and the axons are here. So what we can see is normally the gray area is not that much different. Of course, we can see this may be a little thinner, but the major one is the white. We call it white matter. We can see here. Normally, it's so wide, it's so broad, and this one is very thin. Here is we call it corpus callosum, and connect both hemisphere. You can see this is so thick, and this one is almost gone. Meaning, the patient, the major pathology is axonal degeneration, even though the neurons degeneration is also there. And we can say the axonal degeneration, and this is the corticospinal tract. Here's corticospinal tract, meaning this is from transmission the signal from upper motor neuron to lower motor neuron, and this is degenerated. Degeneration, we say this is become white, is pale, myelin scan, and also this is a sensory column. We can say as a white patient, a lot of patients they were diagnosed as motor and sensory, so that's so-called Charcot-Marie-Tooth disease. So that's why white patients they have different diagnoses. And a major pathology we have never seen before is we can see there's this type of inclusion body we have never seen in any other disease, and this is substantia nigra. So that's the If this area is affected, they will develop Parkinson's disease. And we can see the neurons. This is spinal cord. We can see neurons are lost. This inclusion body. This is the inclusion body. It looks like this. The ubiquitin positive, P62 positive, but not TDP43 and the fuse and others are negative. Meaning, this is unusual inclusion body, not reported before. Because we know in ALS, we have skin-like inclusion that is ubiquitin, 100% ubiquitin and the P62 positive, and 97% of them are TDP43 are fuse positive. But this one is different. That still they're ubiquitin and P62 positive, but TDP43 and others are negative. But most importantly, what we can see is these inclusion bodies. These are a membrane. Their membrane structure there. We can see this membrane structure there. Meaning, we know the ALS, the other ALS inclusion bodies, there's no membrane there, and this one has a membrane. Meaning, it's a unique inclusion bodies. So, to identify what is the inclusion body, so we test many antibodies, and eventually what we found is CHGA. That is a cargo in the large dense vesicles. So we can say CHGA is positive, meaning these inclusion bodies may likely to be large dense vesicles. And we can see synaptic vesicle negative, even though it is suspected to be lysosome. Lysosome is negative, but we can see large dense cargo, including CHGA and the substance P, they're positive. So highly suggests this is a new pathology that contains the large dense vesicle. But anyway, this data has not been published. This data have not been published yet. So to see whether this is really large dense vesicle, we tested in the cell culture system. We tested many different cellular organelle markers. They're all negative. And what we can see when we test the large dense vesicle marker, CHGA, it is really indeed is positive. So we tested the other cellular markers, other dense vesicle markers, CHGA, CHGB, and many others. They're all positive. So meaning this protein, the SPG11 encoded protein, is localized to the large dense vesicles, suggest this protein probably affect large dense vesicle function. So then we developed the knockout mouse model, meaning knockout of this gene to see what will happen. We use the CRISPR-Cas9, so generate a allele with 17 nucleotide deletion. And the mice develop disease. There's a motor impairment. What we can see is after 1 year, the mice have the bin walking there's a problem, and the rotor root has a problem. The mouse have a lifespan of 4 months shorter than normal. We can say it's shorter. This is the wild type mice. This one is the knockout mice. And the brain is smaller. It's one fourth smaller than the wild type. This is the wild type. This is the mutant. The pathology almost exactly the same as what we see in the patient. Many inclusion, we can say the different neurons, inclusion bodies. For example, this is the brainstem. This is the motor cortex, and also we can see in the Purkinje cells in the cerebellum. That's why patient will develop ataxia, and also all the others. And what we can say is, yes, this is ubiquitin P62 positive and containing CHGA and also granulin and other large dense vesicle cargoes. This is the image of large image. You can see what you can see is the CHGA positive inclusion in different neurons. This is the soma, and what we can see most of them. This is cerebellum in the axons. this is very impressive. What we can see is these large tensile vesicles accumulated all together. More than thousands of the large tensile vesicles aggregate together. And this is the soma. What we can see is the soma. And this is in the axons. What we can see is the axons with containing the large tensile vesicle inclusion. And they become very thin. Nobody, you can say this is a normal axon. And this one becoming dilated, huge, and fully blocked by these inclusions. And become very thin, and the degenerated axons are degenerated. So this pathology has never been seen in any other disease before. And so to describe this new pathology, because this contains large tensile vesicle, then cause the axonal degeneration, and we propose this one to be called like large tensile vesiculopathy. You can see what we can see is the axon, the large tensile vesicles can be observed in the nerve terminal of the wild type and the mutant, meaning the spartaxin, the protein encoded by this gene, is not thoroughly responsible for the large tensile vesicle trafficking. They can still traffic. That's why the patient develops disease later. Not they can survive for so long time. And also, we can say these inclusions, sometimes they can track like some other organelles, like a mitochondria. And when we use the immunogold, these inclusions is containing CHGA, meaning it's really the large tensile vesicle aggregation and block the axon trafficking. So we can say this is a we call it lipofuscin. Looks like large tensile vesicles, but they're not. There's no vesicle structure, and we can see in these inclusions there lipofuscin structure, but their density is much lower, still much lower than the large tensile vesicles. So this one is so we collaborated with Dr. Mingjin Chen and Dr. Hageman, and we want to see what is happening there in the neuronal property. And what we found is these neurons knock out the mouse neuron, they become very active, and compared to the wild type. This is a common phenomenon for the neuronal degeneration. When the neurons are before they're dying, they become crazy. They fire a lot, and the mechanism is not known why they become crazy. And we can see the activity is greatly increased, and the time per minute of the firing is much increased. And we tested if this one is related with the large tensile vesicle impairment. And we know in the large tensile vesicles, the most prominent cargo, well-studied cargo, is called substance P. And the substance P is very small, so they can penetrate to the spinal cord. So we test that one. We can say this inclusion, the hyperexcitability of the lower motor neurons is related with substance P. When we put the substance P there, it will greatly enhance the hyperexcitability. The substance P, there's a receptor in the postsynaptic membrane. We called pathogenic, so TACL1. And when we put substance P there, it will increase the firing, meaning the TACL1 is increased. So we tested whether it is true. Yes, we can see the substance P receptor is increased. And not only for this mouse model, because the other ALS and the other disease, if they involve the spinal cord, patient will always showing spasticity and also hyperflexia. And that one may be the same. So we tested also another mouse model, SOD93A. It also has the greatly increased hyperexcitability of the lower motor neuron. So this one has probably have a great implication, because if this one increase, if the substance P receptor increase is responsible for the higher firing, because we know higher firing is related with neuronal death. And our Retinol, and that's the ALS treated drug, that is to inhibit the firing. And that seems is effective. So if you use this substance P receptor, tachykinine receptor antagonist, that have the same effect, that will inhibit the higher firing, and may also have the therapeutic effect. And interestingly, the substance P antagonist is available in clinic. That at least there are two drugs called prepitant and litipitant. And this one has been in clinic for more than 20 years. And it is used for treating the patient with the nausea and vomiting when they in the cancer treatment, and has been pretty safe. And if this one is effective, meaning we can use this one. We just repurpose. It's clinically available. If they approved, we can just repurpose for treatment of ALS, because we can see not only the SPG11, ALS1, SOD cause ALS the same. So it's a summary, and this gene is very frequent, and because in our normal population, in our exome and genome sequencing database, there are about 200 people that will have one carrier. So if that is a frequency, we interpret the patient in United States, for example, 330 million, and we have like about 500 people. There are 5,000 people. Of course, these patients, they will live like 2/4 of normal population, meaning they can live for 60 years. So we estimate the patient will have more than 4,000. And in the whole United States, we have around 115 to 30,000. So the SPG15 cause ALS will have 4,000, more than C9, operating frame, and SOD. So it will be the most frequent ALS type. But this disease clinically may not be diagnosed as ALS, because what I said previously, they may diagnose as SPG11. Some patients will diagnose with juvenile ALS, and some of them will diagnose with Parkinson's disease. Some of them will diagnose as ataxia. So therefore, all different clinical diagnoses, but they will really caused by the same disease, but it's the same gene and the same type of disease, just a different aspect of clinical appearance of the same disease. And the second one is we identified this protein is a large score associated protein and affect large score, most likely affect large score trafficking. The third one is maybe this finding, because especially the substance P receptor high upregulation suggests a therapeutic strategy, and that can be immediately tested in clinic. That's the substance P, FDA approved, the substance P receptor antagonist, because they suppress the hyperexcitability. I think I will just stop here and answer some questions.
Questions, Paul?
Great work, HX. Within a family that has the same mutation, does everybody have the same phenotype? Are there some individuals that have a Parkinsonian features, some that have a motor neuron disease features, some with CMT features? Or within a family, is it always the same clinical presentation?
There are some, even the same mutation, the disease onset, they also have some variations. For example, actually most of the mutations, what we can see in the SPG11 and ALS5. They're truncation mutation, meaning the protein is gone. So even though the protein, the full protein is missing, and the disease onset, I think the disease onset is very objective. The disease onset is still variable. Some of the earliest one can be like 5 years old, and some of them will be in the teenage. Some of them they will be maybe even later. But anyway, all of them will develop disease before 25 years old. So that means clinically there are some variations. And the patient with Parkinson's symptoms, if you treat with like dopamine, they will be therapeutically effective. And they put a deep brain stimulation in the ALS5 patient with the Parkinsonian. They're effective, sustainable. Meaning it's really Parkinson's disease. But the cause is the loss of the protein called spartaxin. It's interesting disease.
Next question right here.
Perfect. With the degree of white matter atrophy that happens in these patients, do they have any sort of cognitive phenotype?
Yes. Actually, I think the cognitive one probably is the most prevalent, and I think almost all of them have the cognitive problem.
Right, so I'm curious then, you said that the disease presentation, the onset is very variable in the patients from 5 years old to teenagers and also some adult onset. But if I remember, your mice were almost more than 2 years old before they were symptomatic. So how do you reconcile that?
What when you make the judgment whether the mice has disease onset, and we use for example, the beam walking motor mode. And that one is much less sensitive than the clinical, judgment. Our humans, we walk on 2 legs. The mice, 4 legs. So whether they have problem, they have spastic paraplegia, it's so difficult. Only when they have the severe impairment, then this can be detected and beam walking and the rotor road. So it will be much later. So humans, we only have 2 legs. So when we wobbling, it's much more sensitive than the mice.
Right, so the ALS2 mice have spasticity, the ALS2 knockout mice, and so there are tests available to be able to detect that early.
Which mice?
The ALS2 knockout mice.
No.
Which also is juvenile ALS.
Yeah, I know. ALS mice, we developed ALS2 mice. And when we walk with the rotor rod, and if we use the routine program like 3 minutes to 40 RPM, and we cannot detect. Only when you use the enhanced program, you may detect. So ALS2 is clinically is less severe than ALS15, like ALS5. And ALS5 is more severe clinically.
Pascali's last question.
Yes, have you looked at neuropeptides? Large dense core vesicles are loaded with neuropeptides. Can this explain partly the pathological symptoms?
The load, the neuropeptide, to detect the neuropeptide is the major problem is that the neuropeptide, they are all cleaved to become very short peptides. For example, substance P, right? Substance P. Actually, the processor has 3 peptides. So when they get into the large testicle, when large testicle is maturation, so they're chopped to small 11 amino acids. So this one, they're chopped. So it's difficult to detect whether they are present or not.
All right, great. Thank you. Let's thank one more time.